Welcome to the deep dive. If you ever wished you could hit the pause button on life just for a moment and come back totally refreshed, well, today we are diving into the science that makes that concept far less fictional than you might think.
It's a fascinating area.
We are tackling one of the most fundamental questions in biology. What if we could truly stop, or even, partially reverse the hands of the biological clock?
And this isn't just theory anymore. We are analyzing two phenomenal scientific reports today. The first gives us this detailed molecular blueprint for functional life extension.
And it was discovered in a tiny worm, of all things.
Exactly. It details the precise cellular mechanisms for developmental arrest and reversal. Then the second report introduces a revolutionary, rapid method for measuring these complex aging dynamics.
Across multiple species, right?
Yeah.
Including humans.
Yes, in real time.
So that's the mission for you today. We are going to uncover the biological mechanisms that allow an organism to pause its age. And then we'll get into the new tool, a new kind of aging clock based on fluorescence that promises to track your biological age instantly.
Bypassing days of traditional lab work, it really connects the why of biological reversal with the how to measure it in the lab.
Okay, let's start with the worm, the nematode C. elegans. It's a bit of a celebrity in longevity research for a reason. It has this incredible lifespan plasticity.
He really does.
So what are the environmental cues that trigger this extreme survival strategy? What does this pause even look like at a cellular level?
It's triggered by, classic survival stresses, overcrowding, running low on food, heat stress.
The usual suspects.
Right. And when these factors hit, the worm enters a survival state. It's called the dauer diapause. Think of it as a kind of a developmental cryo-sleep.
So this just halts everything.
It halts development in an alternative third larval form, the L3 dauer. But here's the crucial takeaway. When the worm exits this state, it doesn't just survive the pause. It genuinely rejuvenates.
Rejuvenation is the key word there. It's hard to wrap my head around true functional reversal. I mean, if you put your car in storage for months, you still have to deal with degradation when you pull it out.
That's a great analogy.
So does the research suggest why the damage acquired during that pause isn't permanently written into the organism?
The evidence for functional reversal is powerful. When these post-hour animals resume normal life, they show absolutely no lasting functional decline.
None at all.
None. Their adult lifespan and their reproductive capacity, their brood size, are completely unchanged compared to worms that never arrested. The data shows, for instance, that adult DAF2 mutants who spent 20 days in dauer had the same subsequent lifespan as non-dauer mutants.
Wow, so the damage acquired is functionally erased upon recovery.
Exactly.
So if the worm is functionally rejuvenated, how do scientists actually quantify age during this suspended animation? They can't use the standard epigenetic markers like CpG methylation, which our human aging clocks are based on.
Right, and that's where these cutting-edge predictive models come in. Researchers developed what they call the binarized transcriptomic, or bit age clock.
BICI age.
Yes. And instead of looking at DNA modifications, this clock reads the entire cell's library, the transcriptome. It looks at thousands of genes and simply asks, is this gene on or off relative to a baseline?
So a simple binary snapshot.
A binary snapshot of the cell's internal machinery, yes. And it's so powerful it can accurately predict the worm's biological age and even its remaining lifespan.
That's a highly specific tool. So what did the bit age clock reveal about the pace of aging during the dauer state? Is the clock stopped or just running in slow motion?
It confirmed that aging does continue, but the rate decreases sharply over time. It suggests a progressive commitment to longevity.
Okay, so it's not an immediate halt.
Not at all. If you look at the initial phase of diapause, say, between day 1 and day 4, the biological aging rate was relatively high, around 0.79. But then, as the worm settles in for long-term survival, tracking from day 4 to day 30, that rate just plummets.
What?
To approximately 0.12.
That difference is dramatic. A drop from 0.79 to 0.12 isn't just slowing down. That suggests a fundamental biological strategy switch, one that happens after a few days of arrest.
You've nailed it.
It's like the worm starts by slowing the pace and then flips a major internal switch to enter a true hyper-efficient maintenance mode.
That analogy is excellent. It's a duration-dependent strategy. The organism is actively prioritizing survival mechanisms to slow the accumulation of damage to an absolute minimum.
Okay, let's look at that mechanism switch. What is the molecular blueprint for this dramatic age deceleration? If we could translate the worm's survival playbook to higher organisms, what are the key cellular instructions?
Well, the researchers analyzed the gene expression patterns across the whole diapause state at day 1, 4, 15, and 30, and they identified a highly coordinated survival system built on 3 core transcriptional shifts.
Okay, what's the first component of that system?
The first is basically slamming the brakes, a massive downregulation of metabolic pathways.
So things like,
Think glutathione, carbon, and fatty acid metabolism all going quiet. This totally lines up with what we see extending lifespans in other models, like dietary restriction.
right, you slow down energy consumption and thus the production of damaging metabolic byproducts.
Exactly.
So slow down the engine. But if the engine is running slow, how do they handle the tiny bit of damage that is still occurring, especially over 30 days?
And that leads to the second shift, deep cleaning. There is a significant upregulation of cellular quality control, or proteostasis.
Burritostasis.
Yes, so this includes pathways like the proteasome and autophagy, the cell's recycling, and waste centers. So they aren't just slowing down metabolism, they're simultaneously ramping up the internal cleanup crew.
To actively chew up and mitigate any accumulated protein damage.
You got it.
And the third component, which sounds more proactive, is the genome maintenance priming.
Yes. The third shift is the preemptive defense strategy. They see this transient, nonlinear increase in DNA repair pathways like homologous recombination and nucleotide excision repair.
And when does that happen?
It peaks early around day 4. It's almost like the worm runs a full diagnostic and pre-service check on its entire genome right at the start of the pause, preparing the system for the long haul of quiescence.
So it's preserving its genomic integrity before it enters that long rest period.
Precisely.
Speaking of duration, the source material talks about this really interesting phenomenon called the proteasome flip upon exiting the dauer state. This suggests the recovery process isn't fixed. It depends on how long the worm was arrested. Can you unpack that dynamic?
It's a beautiful illustration of adaptive recovery. If a worm was arrested for a long time, say 15 or 30 days, they had already strongly upregulated those proteasomal cleanup genes during the diapause.
So they've been running a high-efficiency deep cleaning operation for weeks.
Exactly.
So when they wake up, they're already
at excess capacity. They built up so much maintenance reserve that they can actually downregulate those cleanup genes immediately upon exit.
So they relax the system back to a baseline efficiency?
Yes. But the early dauers, the ones only arrested for a day, they hadn't built up that reserve, so they were forced to rapidly upregulate proteasomal genes upon exit to handle the sudden metabolic ramp-up.
I see. So the duration of the arrest dictates the mechanism and speed of functional recovery.
It confirms it, yes.
And linking that functional recovery back to genomic integrity, the sources identify a specific age-related transcriptional damage that is efficiently reversed. Tell us about the gene-like dependent transcription decline, or GLTD.
GLTD. It's typically caused by accumulating transcription-blocking lesions, often seen in aging. It basically makes it harder for long genes to be accurately transcribed.
And the somatic cells in dowers are postmitotic, right? They can't just divide or refresh themselves.
That's the key. The fact that the worm can recover successfully from UV damage upon exit shows they are actively fixing this foundational problem.
So what's the repair crew for that specific damage?
It's the transcription-coupled nucleotide excision repair, or TCNER. This mechanism is critical because it specifically targets lesions in actively transcribed regions of the genome.
And by activating TCNER during the exit phase.
The worms efficiently restore their transcriptional integrity, reversing that age-related decline. This, combined with the other shifts, is the molecular key to genuine cellular rejuvenation, not just slowing damage, but actively erasing it.
That gives us the molecular blueprint. We know the what and the how for this organism's reversal. But scaling that science to higher organisms requires speed and accuracy in measurement.
A huge challenge.
Transcriptional clocks, like the bed age clock we discussed, or even human epigenetic clocks, they're powerful, but they require cell destruction, DNA isolation.
Multi-step preparation, that process can take days.
And that time lag is a massive bottleneck in geroscience research. Imagine trying to screen hundreds of potential anti-aging interventions.
Or track how quickly an individual's organs are aging over the course of a week. Traditional clocks are simply too resource-intensive and slow to track biological age dynamics in real time.
And that necessity led to the second major finding, the development of senescence-dependent fluorescence lifetime imaging. S-FLIM.
S-FLIM is a revolutionary tool because it bypasses all that complex biochemical prep. It doesn't need you to isolate DNA or perform lengthy in vitro PCR.
So it uses advanced imaging instead.
Exactly. It measures a biophysical parameter inside a living cell, and it gets a result with a turnaround time of under an hour and a half. That speed just changes everything for rapid screening.
That speed is phenomenal. But let's get into the mechanism. What exactly is S-FLIM measuring inside the cell that links to aging?
It's tracking a very subtle biophysical parameter, the micropolarity of the nucleolus.
The nucleolus, that's the factory where we produce ribosomal RNA, or rRNA.
That's the one. And as cells age or enter senescence, they exhibit higher ribosomal DNA methylation.
A chemical tag buildup.
Yes. And that tag reduces the production of the precursor RNA, the pre-RNA. This creates a quantifiable shift in the nucleolus, increasing the ratio of mature RNA to the precursor.
And that physical shift changes the surrounding chemistry.
The micropolarity of the nucleolus, exactly. S-FLIM is specifically designed to detect that subtle environmental change.
And that's where the novel dye comes in. We're talking about FLP1.
Yes, FLP1 is a specialized hybrid polymazine dye. It's incredibly targeted. It shows a 410-fold fluorescence enhancement specifically when it binds to rRNA. But the key is its fluorescence lifetime.
How long the light signal lasts after you excite it.
Precisely, and that lifetime is acutely sensitive to those changes in nucleolar micropolarity. We are essentially using the dye as a physical probe to instantly assess the biochemical status of the cell's ribosome factory.
That level of physical resolution sounds powerful for identifying individual aging cells or senescent cells. Can S-FLIM reliably distinguish them?
Highly reliable, and the contrast is sharp. It successfully distinguishes proliferating, healthy cells, which show a fluorescence lifetime greater than 2.0 nanoseconds from senescent cells.
Which drop below what?
They drop below 1.0 nanosecond in various human cell lines and even mouse kidney tissue.
This precision must make it a powerhouse for screening new therapies, particularly those aimed at clearing senescent cells, the senolytics.
Oh, absolutely. It showed superior performance for tracking therapy-induced senescence, or T-type. For instance, S-FLIM could identify early senescence after just 24 hours of adipose side treatment.
And the traditional method couldn't.
The traditional beta-gal method failed entirely. What's more, the study noted S-FLIM offered a contrast ratio 10 to the 19th power higher than older intensity-based methods.
Let's focus on that contrast ratio, 10 to the 19th fold. That is an enormous number. What does that kind of staggering contrast mean practically for a researcher trying to track minute changes in aging?
It means the system is virtually impervious to the noise and variation that plague older methods, things like changes in probe uptake or cell size. It's seeing signals that, to previous technology, were just static.
So you get highly reliable, sensitive data on cellular aging status.
Independent of all those other physical factors.
Now, let's bring it up to the whole organism level. Can S-FLIM generate a predictive biological clock for mammals, much like the Beatty age clock did for the worm?
Yes. Researchers focused on neutrophils, a type of immune cell that's easily accessible through blood samples, to create organismal clocks.
And in mice?
In mice, the FLP1 fluorescence lifetime in neutrophils decreased linearly with age from 2 to 20 months. And crucially, this correlation was exceptionally strong, an R-squared value of approximately more 0.97.
An R-squared of 0.97 means that the S-FLIM clock is tracking the mouse's biological age almost perfectly. That's a massive technical achievement for a biological aging measure.
And this high correlation held true for humans. In volunteers ranging from 18 to 73 years old, the lifetime measured in neutrophils also decreased linearly with chronological age.
What was the R-squared there?
Approximately 0.91. So this confirms S-FLIM is a viable, rapid method for real-time aging rate assessment in humans.
And we have concrete proof it can spot premature aging or longevity phenotypes. Give us those examples.
So in the premature aging model, young 2-month-old mice were treated with the chemotherapy agent doxorubicin. They exhibited a neutrophil lifetime of 1.1 nanoseconds.
And what would that be equivalent to in a naturally aged mouse?
That reading is functionally equivalent to the lifetime of a naturally aged 15-month-old mouse, which was 1.0 nanoseconds. S-FLIM instantly flagged the acceleration of biological age caused by the drug.
And on the flip side, the ultimate validation, measuring the longevity phenotype of the C. elegans, DAF2 mutant we talked about earlier.
Right. The long-lived DAF2 mutant showed a much flatter S-FLIM aging curve than the normal wild-type worm. The nucleoli lifetime of a 19-day-old long-lived mutant was equivalent to a wild-type worm that was only 7 days old.
And what was the reading?
Both measured approximately 1.92 nanoseconds. S-FLIM accurately quantified the functional youth of the long-lived strain in real time.
So let's synthesize these two extraordinary findings for you. The learner. We've seen the molecular blueprint, this profound realization that genuine cellular rejuvenation is possible, driven by coordinated metabolic repression, enhanced cleanup, and preemptive DNA repair.
And we now have S-FLIM, a tool that provides an instantaneous, sensitive, and real-time measure of those very aging dynamics, moving from single-cell resolution all the way up to highly predictive clocks in living humans and mice.
This confluence of the why, the cellular mechanism of reversal in the worm and the how to measure it quickly with S-FLIM, is fundamentally changing the speed at which geroscience can advance. We're moving from needing months or even years to assess an intervention's effect to potentially getting a highly correlated readout in an hour.
That speed and precision enables things we just couldn't do before. We can screen candidate interventions rapidly, we can fine-tune drug dosages,
And potentially even track the effectiveness of personalized lifestyle interventions on a near real-time basis.
which moves us toward truly tailored longevity strategies.
It makes the future of personalized longevity interventions feel much closer. And that leads us to our final provocative thought for you to chew on, built on S-FLIM's capabilities. We know different organs within the same human body age at different rates. So given S-FLIM's ability to generate highly correlated, tissue-specific aging clocks and rapidly assess therapeutic efficacy, what questions will this tool allow researchers to answer about the variable aging rates between your liver, your heart, and your immune system?
And crucially, what will that personalized, organ-specific data mean for designing clinical intervention trials that target aging in the specific tissues that need it most? That's the challenge ahead.
We'll catch you next time for the next deep dive.